Hi, I'm Sarah Spencer and I'm an Associate Professor at RMIT University. Today I'm going to be talking to you about some of our work in harnessing the brain's immune system to improve memory. So I'm sure all of you are very familiar with having been sick at some point in your life, and you will be aware of having encountered a bacterial or viral infection. And probably personally familiar with the suite of symptoms that come about as a result of being sick with a bacteria or a virus. These include things like withdrawal, so you're perhaps not so willing to socialize, include lethargy and sleepiness, mood changes, generally in the direction of depression. They can include enhanced pain and a disinclination to eat. They can also include stress activation, and sometimes you will get a fever. This suite of sickness behaviour is usually or often accompanied by an increase in circulating factors that can be measured in the blood, as well as occurring in the brain, some of which are known as cytokines. And in addition to all of these different behaviours that are associated with sickness, we also sometimes see cognitive impairment, and this is what I want to talk to you about today. Now, we know that this suite of sickness behaviours can occur in response to a bacterium or a virus. We also know that these types of sickness behaviours can occur in response to lifestyle factors, in particular in response to diet. And we know that the early life period is really important for programming our sickness behaviours long term. And so we see that perinatal diet is really important. We also see that adult diet or diet throughout your life is really important, and in combination with obesity can also program this chronic low-grade level of these sickness behaviours. And in addition to this, there is a particular vulnerability during the ageing phase of life in which we're vulnerable to these sickness behaviours. Together, what accounts for these sickness behaviours that we experience is inflammation, and this is the cornerstone of what I'm going to be talking about today. So we've seen in work that my group has been doing that early life poor diet, including early life overfeeding, can lead to brain inflammation. And we've seen that this brain inflammation is associated with cognitive deficits, so deficits in the ability to learn and remember. Now, this is a test of recognition memory. So the subject is asked to recognize an object it has seen before in comparison to an object it has not. And we see that subjects that were overfed as neonates in that very early period of their lives are perform much less well in this test of recognition memory. We also see a similar effect in tests of spatial working memory. So this is a task that's very similar to being asked to remember every day where you park your bicycle. You ride your bicycle to work every day, you park it in a different parking space, and you need to update that memory every day so that each day you're looking for today's parking space and not yesterday's parking space. And we find that subjects that were overfed as neonates are really poor or really inefficient in learning a comparable task to this. In another example, we see that brain inflammation, in particular associated with a high-fat diet, is also a factor in aging. And this is a high-fat diet that we gave in these experiments that is a very short duration. So it's a little bit similar to a period of binge eating over a holiday period, for example. And in this study, we tested our subjects in a test of aversive memory, so a negative situation. Now, aversive memory is really important. You want to be able to remember something that you encountered that was unpleasant so that you either don't go into that situation again or so that you can handle it better when you do. And we find that in these experiments that if you're a young subject, being fed a high-fat diet just for a very short period of time doesn't do anything much to being able to remember this aversive negative context. If you happen to be an aged subject, this also doesn't do anything very much. The aged subjects are perfectly capable of remembering this aversive context. But if you happen to be aged and being fed a high-fat diet, you can see that these subjects are much worse at being able to remember this aversive context. So this brain inflammation that occurs with aging and high-fat diet is also associated with this cognitive dysfunction. So what do we mean when we talk about brain inflammation? Well, one of the main factors that my team has been working on is these cells called microglia. And these pictures are all examples of microglia that have been generated from my group. And you can see in the top panels, microglia in their native state. In the middle panel, you can see a microglial cell that's eaten up or phagocytosed these microbeads that we've given it. So they take up debris and particles from around the brain. The microglia are a major immune cell population in the brain. They are known to be important in development for sculpting brain networks and dictating the brain that is to become. And we know they're also important throughout life in responding to pathogens and injuries. So they respond to those bacteria and viruses that I talked about at the start. Research is starting to come through, though, that these microglial cells actually have important roles in the healthy brain under unchallenged circumstances, so just in day-to-day life. And that's what my team is interested in investigating. And so we're interested in the question of if we get brain inflammation or activation of these microglial cells in the context of cognitive dysfunction, does this mean that this microglial cell activation is leading to the cognitive dysfunction and is a causative factor? Or does this mean that this microglial cell activation is actually acting to countermine or reverse the cognitive dysfunction that's being caused by something else, and that in the absence of these microglial cells, we would actually see worse cognitive function? So this is something that we wanted to answer the question of. Generally speaking, in science, one of the very, very traditional methods for understanding something in any kind of scientific experiment, whether it be your protein of interest, your cell of interest, or your brain region of interest, is to take that thing away and observe what happens to the brain or to the system in the absence of that thing. And that is essentially what we have done with microglia. So we have our subjects that are normal subjects with normal microglia, and we compare these to subjects that don't have any microglia. So we have a transgenic model where we remove microglia from the system. And when we do that, we can study the system in the absence of microglia. So you can see that the microglia in this picture are the green sort of blobby kind of cells. And you can see in the second picture that they're pretty much gone from throughout the brain. So they're depleted to by 95% throughout the brain. And when we do this, it was actually when we first started doing these experiments, it was really quite surprising that not particularly much happened. So these cells account for about 10% of the total brain volume, and not really very much in the way of earth-shattering findings happened when we first take these out. So the subjects don't get sick. They don't display a stress response or behavioural indices of being stressed. They don't seem to have any deficits in fertility, at least not with respect to ovarian health. And they also don't have any deficits in memory, which seem to speak for the absence of a role of microglial cells in normal day-to-day memory, right? So if we again conduct this test of recognition memory, where the subjects are asked to recognize or remember an object that they've seen before in comparison to an object that they haven't seen before, we see that these guys with the microglia gone perform just as well as the naive animals with their microglia intact. However, in the course of doing these experiments, we noticed that the microglia spontaneously repopulate the brain. We take the microglia out, and by about 7 days later, they are spontaneously coming back again. And when they do this, they look a little different. So normal microglia in a naive subject have a nice round cell body with lots of ramified complex projections coming off. And these projections are the means by which they survey the brain to figure out what's going on, essentially. When we deplete the microglia and then they spontaneously repopulate, they have projections that are much less complex and cell bodies that are much bigger. And so this shape or morphology is more similar to what we would normally see under an activated microglia situation when the microglia are responding to a pathogen or to an injury. So we wondered whether this might affect not only the microglial behavior, but the behavior of the subjects. And so we found that, again, when we run this recognition memory test, these subjects actually perform better at recognition memory when the microglia have been depleted and have now repopulated the brain and have undertaken this different kind of shape or morphology. We also, just to verify this wasn't a spurious finding with respect to this recognition memory test, we also tested them in a place recognition test, which is more similar to the idea of recognizing when an object has been placed in a spot that it's not supposed to be, such as if you put your keys into the fridge. And again, we find that these subjects are much better at this test, at recognizing that this object has been moved, if the microglia had depopulated the brain and then repopulated. So how might these microglial cells do this, and why might microglia be involved in cognition, and why is cognitive function better when they repopulate? So for a long time, we've considered the unit of cognition to be the neuron. So the neurons in the brain are the cells that communicate with each other using electrical signals. They're thought to be activated when a stimulus occurs to create a memory, and the connections between neurons become stronger in accordance with that memory. We now know that neurons don't act all by themselves, and they act in the context or the environment of a whole lot of other cell types, one of which is microglia, which is represented by these blue ones here. Another of these is astrocytes, and I've got a picture of astrocytes, these nice bright green ones down the bottom here, and I'll come back to astrocytes a little bit in a minute. But we thought that maybe in the context of microglia depleting the brain and then repopulating the brain, these microglia are in a place to interact more with the neurons and strengthen the connections between those neurons to strengthen the memory. So this picture is a segment of the neuron. This is a segment of a dendrite. And you can see these little hairs coming off the sides of it. These are the dendritic spines, or sort of the business end of where one neuron connects with or talks to another neuron. And these are strengthened or matured in accordance with creating a new memory. You can see down here you've got a number of different shapes of these spines, and we can see one here that is a bifurcated spine. And this is one of the more mature shapes that these spines take on when the neurons are talking to each other. And this is just a reconstruction of that dendritic segment there. And we can see that when the microglia have depopulated the brain or been removed from the brain, and then they repopulate the brain, we get more of these bifurcated or mature synaptic spines that potentially reflect a stronger connection between the neurons. So we thought that maybe the microglia might be in a position to be interacting with or essentially sculpting and shaping those spines, so nibbling away at the spines to make sure that they mature in the same way. So we tested the capacity for microglia to do this, and we actually found that there wasn't too much difference in the way the microglia were behaving in the sense of being able to consume foreign particles or reflective of their potential capacity to interact with that spine and reshape it. But I mentioned astrocytes before, and at the same time as the microglia have been gone from the brain and then they repopulate, and we find this improvement in memory and we find these strengthened or more mature dendritic spines, we actually see that astrocytes are taking up more of these introduced foreign particles, potentially reflective of astrocytes being more able to interact with the neuron and shape these connections between the neurons. And in accordance with this, we find that there is an increase in astrocyte density in the brains of these guys that had their microglia depleted and then repopulated. And so what we think here is happening is that when we deplete the microglia, they're gone from the brain, then they repopulate the brain and they look a little different. And we still believe that this change in the shape of the microglia is reflected in a change in behavior. We just haven't been able to yet identify what that behavioral change is in the microglia. But we do see that astrocyte density is increased and astrocyte activity is potentially increased. So together, the changes in these two cell types potentially lead to a strengthening of these connections between the neurons when a memory is formed, and a strengthening or an improvement in cognitive function in this regard. So we're definitely still working on this question. I just wanted to point out to you that we're also working on a whole host of other questions with regard to microglia. I mentioned at the start that we're looking at the role of microglia in the perinatal period and how this might program the brain long term. We're also working on the role of microglia in mood. We have some collaborators that have a strong interest in the role of microglia in pain. We've done a little bit of work on microglia in stress. We've been able to determine that microglia and corresponding circulating immune cells probably don't impact on female fertility too much, but they likely do impact on healthy aging. We're also doing some experiments to look at microglia and some equivalent cell types in the gut. And we have preliminary evidence to suggest that microglia may drive certain aspects of circadian rhythms, and certainly microglia are involved in regulating hunger and satiety. And to top all of that off, we have shown that sex hormones are able to influence how microglia function. And so we're still working on a whole range of projects with these questions in mind. And finally, I'd just like to say thank you very much to my team members, in particular Luba Szymanski, who's an invaluable postdoctoral fellow in the group, and all of the students. I'd also like to mention Simone DeLuca, who did a lot of the characterization of the model that I spoke about, and of course the funding bodies. Thanks very much.